Hello, welcome to another video from the conscientious biologist, Ben Gallagher. This one's on blood glucose regulation, and then we tag on diabetes at the end of that. Blood glucose regulation is one of the key homeostatic mechanisms or examples of homeostasis, but this one is a hormonally controlled example of homeostasis. So this video very much follows on from the last one, which looked at the hormones and the endocrine system. If you haven't watched that one already, I would advise you to do so, but this video does function as a standalone video just on blood glucose regulation as the homeostatic process. It's from the GCSE syllabus, and as always, please do subscribe to my YouTube channel if you haven't done so already. Thank you. So as I just said, blood glucose regulation is an example of homeostasis. So let's quickly recap what homeostasis is. It's this maintenance of a constant internal environment. It works by negative feedback loops, and that's to maintain balance. So this is the example we looked at a couple of videos ago, whereas if you've got someone stood there, they've got kind of a center line as to where they should be. If they lean too far in one direction, then to correct that they've gone too far one way, they have to push back in the opposite direction to get them back to the balance point. But of course, if they push too far, they've now got an issue that they go too far the other way. And to correct that, they need to push back, do the negative of what happened to put them back on balance, because that will take them back to where they were. But if they push too far, they'll go back too far right again, and so on and so on. That's how you keep balance. If you right now quickly stand up on one foot, you'll notice that whilst you may appear quite still, concentrate on what's happening on the bottom of your foot, and you'll feel there's constant adjustments and movements as you push to the right to correct or negate. If you're far to the right, you push to the left to negate that, and so on and so forth. It's this negative feedback system to keep things on balance. So that's a negative feedback loop over there. And homeostasis is all about maintaining balance, and that all systems must respond to imbalance by doing the negative of the thing that took them off balance. That's a negative feedback system, and it's how homeostatic systems maintain a constant internal environment. We're now going to look specifically, though, at blood glucose regulation. Now, there's a lot of biology in the explanation of blood glucose regulation. This is why it's a favourite with examiners, but a lot of students really struggle with this, because to correctly explain and answer questions on blood glucose regulation, you need to pull information from loads of other areas of your course. So we're going to start with this really important question of why regulate the level of glucose in the blood. Okay, so think about why that glucose is there. All cells require glucose for respiration, so they can get the energy from it. So this is why we need glucose in our blood, because that energy powers the cell's metabolism, and remember, metabolism is life. So the glucose level in the blood should be enough to supply this need without getting too high or too low. But that still begs the question, why can't it get too high? Why can't it get too low? Wouldn't it be sensible to have the blood glucose level always really high, because then no matter how much the cells need, they can always pull it from the blood? But actually, that's really dangerous. So let's look at the two scenarios of it being too high or too low. Okay, the big worry with glucose is that it's a solute, and if it's a solute, it's going to affect osmosis. So this is where you need to pull in your cell biology and your osmosis and your transport through membranes knowledge. I said you need lots of areas from the course. So if the blood glucose level was too high, so on this diagram, that's just a bit of a blood vessel. So that could be a bit of capillary or arterial vein, it doesn't matter. But the pink shading inside, that's the blood. Now, if I put loads of little yellow dots on there, those yellow marks are glucose molecules. So there's a high glucose concentration in that blood vessel. Now, if there's a high glucose concentration, then there must be a low water concentration, because think, your blood is a solution. It's got plasma, the liquid or water element, and it's got other stuff, red blood cells, white blood cells, platelets, hormones, glucose. Okay, they're all solutes that affect the concentration of water. If you've got loads of glucose, then it means you've got less water relative to the cells that are around it. So think what will happen. If I've got blood with a relatively low water concentration, then water is going to move into the blood from the surrounding cells by osmosis. If you've suddenly got lots more water in the blood in the same amount of space, you're going to get high blood pressure, which is very dangerous. We talked about it on a previous video in disease, but you could get blood vessels literally bursting around your body, meaning they're not supplying blood to the area that they should be, which is what can cause heart attacks and strokes and loads of other problems. Okay, so you don't want your blood glucose to go too high because of this. It acts as a solute, which is going to lower the water concentration. But flip that round, you also don't want a low glucose concentration in the blood, because if you've got a low glucose concentration, then the water concentration is much higher. If the water concentration in your blood is quite high, water will move out of your blood, so it's going to cause low blood pressure. If you've got low blood pressure, you're not going to have enough pressure to send it up to your brain, to send it everywhere it needs to go, and that's bad for the body as well. So in both those scenarios, it's bad news for the body, be it a high or a low blood sugar concentration, and it all comes back to osmosis. So please, any question in the exam that comes up about blood glucose, always be thinking, can you get osmosis into your answer? Now, if we don't want all that glucose in the blood, but we've got loads of glucose because we've eaten food, we've digested the carbohydrates, we've absorbed the glucose through our small intestine, all that glucose is now in the blood. If we don't want it in the blood, then we need to store it safely somewhere. So let's now look at this question, how is the glucose stored? Well, when blood glucose level is too high, it must be removed from the blood and stored somewhere else than the blood. But when the blood glucose level goes too low, you need to be able to get it back from those stores and put some back into the blood. In other words, when you eat and you put all that sugar into your body, you don't want it in your blood, so you need to pull it out of your blood and store it somewhere safely where it's not going to do any harm, where it's not going to mess with osmosis. But when you go low, you need to be able to open those stores up to get some of it back. Okay. Now, glucose is stored mostly in liver cells and muscle cells. So link this to the last video. Those must be the target organs for the hormones that control this. Now, let's look at a couple of scenarios here. If I put up a cell, and let's just say this is a liver cell. If I know that's the cell that's going to store the glucose, for simplicity, let's get rid of all the organelles from inside it, and let's put that glucose in there. So this cell has now absorbed all that glucose from the blood. Now, because glucose is soluble, it makes the cytoplasm of that cell into a sugary solution. Remember, glucose is a sugar. Because it's now a sugary solution, it has low water concentration. We've got the same kind of scenario as when we had too much sugar in the blood. We've now got too much sugar in the cells. So with too much sugar in this cell, water's going to move in, which means that cell's going to swell up and it's going to burst and it's going to break down. Well, we can't have our liver cells all swelling up and exploding. That wouldn't be any good at all. So let's look at a better scenario. I'll leave that one over there so you can see that when the glucose is just dissolved in the cytoplasm because it's soluble, it's a bad scenario because water will move into the cells. But on this cell here, instead of having the glucose all dispersed throughout the cytoplasm, what the cell can do is take that glucose and it can stick the lumps together like this. That's called polymerization, when it forms a polymer. And if you make a polymer of the glucose, the polymer is insoluble, meaning it's not dissolved in the cytoplasm, meaning it doesn't affect the solution. So glycogen, which is just that polymer, it's still glucose. Look, it's exactly the same yellow lumps. But all I've done is, instead of taking those yellow lumps of glucose and have them spread out, I've stuck them all together. The stuck together lumps of glucose is called glycogen, but please don't be thinking glycogen is some totally different thing. It doesn't convert the form. It literally sticks glucose together in a lump. It's like having lots of Lego bricks, or you can stick them all together in one piece. If I held up loads of loose Lego bricks in this hand and a lump of them stuck together in here, they're still Lego bricks, but that's all stuck together. Those are all individual. It's exactly like that, with glucose all spread out over in the first cell. In this nearer cell, they're polymerized into this insoluble glycogen, which means it doesn't affect the water concentration. So no extra water is going to move into that cell, no extra water is going to move out. And also, because glycogen is really compact, look, you can stuff it all away in the corner. It can be stored safely and efficiently when it's in this polymer form. So really, for blood glucose control, and this is what we'll look at on the next slide, What you need to do is get the sugar out of the blood, get the glucose out of the blood into the cells and locked up in glycogen as quickly as you can. When the body needs more glucose, you're going to need to break down that glycogen, literally break the bits off to put some more glucose back into the blood. It's that straightforward. But let's move on from here and look at the negative feedback loop for how that actually works. So this is the negative feedback loop that we're going to look at now. It's just like the one we did for thermoregulation. You want your glucose to be at a relatively constant level, but of course it can go too high and it can also go too low. And it's about keeping that balance. Just like when you're stood, you can go too far left, you can go too far right, but you're trying to keep a balance in the middle. Now, if you go too high, the endocrine gland that controls it is the pancreas. And if blood sugar is too high, the pancreas secretes means releases, secretes insulin hormone. That insulin hormone will go into the blood, go everywhere in the body, but the target organs for insulin are the liver and muscle cells. And what they will do in response to being stimulated by insulin, so they will have receptors on their cell membranes, insulin hormone will lock onto them, and those cells will go, oh, stimulated, grab all the glucose out of the blood and convert it into glycogen, get it stored just like we saw on the previous slide. Now, if they do that really effectively, the blood sugar level might get too low. So you need to flip it back. You need to negate that. Again, it's the pancreas, but the pancreas releasing a second hormone, a hormone called glucagon. Now, my students have always got confused because we're talking about glucose, glycogen, and glucagon. They're all very similar. You just need to make sure you know which one is which. Glucagon is the hormone. Okay, so the pancreas secretes glucagon. Again, the target organs are the liver and muscle cells, but this time they break down their glycogen that they've built and start to put some of the sugar back into the blood. But of course, if they put too much, the level goes too high and we go round and round the loop again. Just like when you're balancing, you're never still, you're constantly adjusting. This is what your body's doing. The pancreas is constantly going, oh, it's gone a bit too high, a little bit of insulin. Oh, it's gone too low now, a little bit of glucagon. Constant little tweaks and adjustments to keep you on balance. Now, to summarize this in a table, like we saw on the previous videos, there's 4 key things you need to know: the name of the hormone, where it's produced, what its target organs are, and what its instruction or its message is. So for this, I've summarized it here. The insulin, well, they're both produced by the pancreas. Target organs are the same, muscles and liver. But the instruction of insulin is to take in glucose and convert it to glycogen. Glucagon is the reverse, break down your glycogen and release the glucose. Now, I should say insulin does actually have a third target organ, but it's not really one that we need to know about too much. But the third target organ is your fat cells, because insulin also tells fat cells to take in glucose and convert it into fat as a long-term store. So actually, most people who are overweight and have too much fat on their body, it's not necessarily because they've eaten fat foods or fatty foods, it's because they've eaten too much sugary foods and carbohydrates, and their body, in response to insulin, converts the sugars into fats, okay? But that's an excess thing. Your natural response is this, to store sugars as glycogen ready for when you need them next. Now, to pull this up in a graphical form, because this is how they love to give it in the exams, if we're looking at the changes in blood glucose level over time, it should be at a constant. So I've put this dotted line on there. But what you'll actually get is something like this if you measure your blood glucose level throughout the day. And in the exam, they'll probably ask you to comment on some of these reasons. Well, look at the massive peak over at the start over there and think about what could cause that sudden increase in glucose concentration. Well, that should be pretty obvious. That's when someone ate some food, they had breakfast or something. If you eat, you digest it, your blood sugar level is going to go up. So that accounts for the rise. Now, if you look on the graph, that rise then steeply drops. Well, that means that near the top of that first peak, the body released insulin. That caused the muscle and liver cells to pull the glucose out of the blood. So the blood sugar level significantly dropped. If you look at the graph, it dropped way below what it should because too much insulin went in to combat that huge spike. So at the bottom just here, look, the lowest point near the start, That must be where the body went, oh, too much, release some glucagon to get some glucose back in. But it was a bit too much, so it went a bit too high, so a bit more insulin, bring it back down, and then it could do the fine little tweaking again. But then we had another drop, okay, pretty much up in line here, okay. We had another drop there. Well, maybe that's where the person just did a little bit of exercise. Exercise meant their cells burnt more glucose in respiration. They pulled that glucose from the blood, so the blood glucose level dropped. So in response, pancreas released glucagon. That put a load of sugar into the blood, which is why it went back up here. But then too much went into the blood, so you released insulin to get it back down and then send it back to balance again, okay? It should be fairly obvious what's going on that graph, but that's what you're likely to be asked to explain in the exam. They might mark anywhere on that graph with like an X and say, explain what's happening at point X. If the level's going back down, you would need to say, blood sugar level went too high, so the pancreas secreted insulin, which told the muscle and liver cells to take up the glucose and convert it into glycogen for storage. Okay, that's the type of answer you need to give. So take a screenshot of this because this basically summarizes everything on blood sugar regulation. But please remember, you've got all that osmosis stuff. You've also got the kind of biochemical links about polymerizing glucose. So there's lots of information on this, which is why it's a favorite in the exams. Okay, last thing in this one that I've just kind of tagged onto the end here is to talk about diabetes. Now, diabetes is a condition whereby the sufferers have an inability to control blood sugar levels naturally. In other words, that last slide, that negative feedback loop, they can't do that properly. But there's two forms of diabetes. There's type 1 diabetes and there's type 2 diabetes. In type 1 diabetes, it's a genetic condition. In that form, patients cannot create functional insulin hormone. Either they don't have the correct cells in the pancreas to make it, or they make the protein wrongly. The gene for insulin isn't correct, so the protein they make isn't the right shape, so it can't lock into the receptors on the muscle and the liver cells, so it just doesn't function. Whatever the reason, type 1 diabetics cannot make functional insulin. That's the most important thing to remember. Now, if they can't make it, that means their blood glucose levels could get dangerously high because they struggle to bring them back down. So it's treated by injecting with insulin. Diabetics would have insulin with them, normally now in sort of pens that you can control it. Some people have implants that can monitor their blood sugar levels for them and act almost like an artificial pancreas. But however they do it, they need supplements of insulin because they can't make their own. That insulin does exactly the same job as human insulin does, but of course they can't monitor the levels internally, naturally, and control it as well as a non-diabetic could do. Okay. You can also control type 1 diabetes, of course, just by having a sensible diet. Don't eat too much sugary food. Don't eat loads of sweets that are going to put your sugar levels up through the roof. Okay, so that's type 1 diabetes. It's genetic, where you can't create functional insulin. Type 2 diabetes is massively on the increase, very sadly, and it's a lifestyle associated disease. In other words, it means it's almost entirely preventable, meaning people develop type 2 diabetes through things that they have chosen to do through their life. Now, they won't have made those choices going, I'm going to choose them anyway and get type 2 diabetes. But it's a lack of education, people perhaps not being aware enough of the risks they're putting themselves at through certain lifestyle choices. So what you've got in type 2 diabetes is where the body stops responding to insulin due to excess overuse. If someone's constantly eating sugary foods and their blood sugar levels are always high, their pancreas is having to make loads and loads of insulin, constantly telling the liver cells and muscle cells and fat cells to take that glucose in and store it. Well, eventually, those cells just stop listening to it. It's as if I was saying to you the same thing over and over again. If I was saying constantly, stand up, stand up, stand up, stand up, stand up, hey, stand up, please, stand up. Eventually you go, oh, shut up, I'm not listening anymore. That's basically what happens in type 2 diabetes. Because the body is constantly going, take the insulin out of the blood, take the glucose out of the blood, take the glucose out of the blood, take the glucose out of the blood. Those cells that can do that just stop responding to it. And as I said, this is largely due to a poor diet, too much sugary food, too much carbohydrates, chips, pizza, burgers, cake, sugary drinks. Ketchup and sauces have huge amounts of sugar in them. Sweets, chocolates, all those kind of things, huge amounts of sugar in the carbohydrates going into your body, constantly elevating your blood sugar levels and requiring a huge amount of insulin. your body just stops responding to that. Now, the treatment for that is still really about injecting insulin, okay? But you'd need a different form of insulin to what your body creates, but one that can still hopefully create a similar response in your muscle and liver and fat cells to try and trick them into doing the same job that they were ignoring before. So it's kind of like going on the please stand up, stand up, stand up. Instead of me saying that, if I said stop sitting down, hopefully you'll do the same thing. I've just phrased it in a different way. You're using a slightly different protein to get the same response. But of course, the main thing is to sort your diet out. If you're significantly overweight, you've got lots of junk food in your diet, there's a serious risk of type 2 diabetes. You can reduce the risk of getting that type 2 diabetes by sorting your diet out. And if you've got type 2 diabetes, you can dramatically improve the impact it has on your life by sorting out your diet. But the key things I'll ask about in the exams are the things I've put in bold. What are the causes of both type 1 and type 2? One is that you can't make the insulin. One is that your body stops responding to it. Let's take a screenshot of this. It's not a lot of information, but it does come up quite often. So from here, we move on to probably the most complex thing in the entire GCSE scheme, which is the hormonal control of the menstrual cycle. It's loads of interactions and cascades of changes that go on throughout the female body on a monthly cycle to control egg release and making a hospitable environment in the uterus for a baby to start to grow. So it's quite a complex video. That's the one I want you to head to next, because it's a big part of the hormones and endocrine syllabus. As always, please do subscribe to my YouTube channel if you haven't done so already. Give this video a like if you find it useful and informative, and do head over to my Facebook page where there's lots of extra supplementary biology just to boost your general knowledge of the subject. Thank you.